Welcome to the class. In this class, we have going to see about the gene isolation, synthesis, and the cloning of DNA. So, especially in DNA extraction, it was followed with taking the sample and going through the process of extraction, and then we are finally getting about the DNA pellets. So, for this, we have to take the raw material, that is leaf material, into small pieces, and we have to cut it down and we have to sterilize it using distilled waters and 70% ethanol followed by distilled water again. So we have to grind the leaves using pestle and mortar with liquid nitrogen, and the same sample, 1 ml of this liquid sample with 3 microliter of beta mercaptoethanol was in 2 ml Erbendorf. We have to take it and vortex it for 15 seconds. This is because this mercaptoethanol will um, degrade the cell walls, and you have to incubate it in the water bath for 60 degrees Celsius for 30 minutes. Then they add the equal volume of chloroform as well as the isoamyl alcohol in 24 part is 1 parts mixed with inversion. So these have to be centrifuged into 15,000 rpm for 10 minutes, then separate the liquids and also the supernatant and discard the supernatant and take the it centrifuge with overnight minus 20 degrees Celsius. Again wash the pellet with ethanol, and 2 microliter of ammonium acetate is added by the inversion. Again we have to centrifuge it, and again we have to add 500 microliter of 96% ethanol, and again we have to centrifuge it for 10,000 rpm. After all, we have to discard the supernatant, and the final pellet will be available in the pellet form that will be white tinge of pellet of DNA can be visible. That should be dried in the vacuum or air dried, and add the TE buffer at minus 20 degrees Celsius and allow the time for suspension. So this is the nutshell, and first thing in the DNA extraction process, the sample should be collected and the cell lysis will be done using detergents or the ST soaps, or else and removal of proteins and other contaminants using protease and other denaturation substances can be added. Then the DNA precipitations will be occur due to adding of alcohol, and the DNA should be washed thoroughly. Then ammonium acetate is added by the inversion or the centrifugation. Again the DNA resuspension will happen, the supernatant will be discarded, again the pellets will be extracted. In case of RNA extraction, it is done by the trisol method. It will be done in a very sterile condition. The homogenization will be happen. The supernatant will be collected. There will be two phases. One phase is separation and the precipitation phase. The supernatant will be discarded and it will be taken for the isopropanol precipitation. Then the pellet cleaning and resuspensions will happen. But DNA can be done in the normal laboratory condition, but it should be done, RNA should be done in the sterilized conditions. So the lysis buffer, trisol or guanidinium thiocyanate when be added, it will inactivate the RNAs present in the air and the surrounding compounds. The trisol or phenolchloroform method, using that we are precipitating and washing, and the elution will be done in the final phase. So these are all the extraction and the isolations of the genes from the plant extract. After that, the genes will be went for the polymerase chain reaction, where the DNA is extracted and amplified into n number of copies. Then restriction, digestion and cloning will happen. So this is the gene synthesis using PCR method. Thank you.